Welcome, everyone. We are delighted to have participants connecting from across the globe, and we'll give it another minute for folks to join, and then we'll get started. I can see people still logging in. All right, so let's get started. My name is Sanchita Bhattacharya, and I'm the Scientific Outreach Program Lead at Import. It's a pleasure to have you all join us for this webinar series. Before we go to the main presentation, I would like to cover a few housekeeping items. The presentation time is 45 minutes, followed by 10 minutes for Q&A. Throughout the session, we encourage the participants to ask questions using the Q&A feature. And we'll try to take a few questions live after the presentation. And this session is being recorded and copy will be made available at a later time for reference. So let's now move to the presentation segment. Today's webinar will introduce the Immune Signatures Data Resource, a large-scale standardized compendium of systems vaccinology datasets. This is developed by the NIAID Human Immunology Project Consortium, HIPC, and as a part of the National Artificial Intelligence Research Resource. So we are pleased to have guest speakers with us today who will present an overview of the resource and guide you through how to access and use the data for your research. So it is now my pleasure to introduce our guest speakers, Dr. Joann Arce. Joann is a systems immunologist specializing in infectious disease and vaccines. She is an assistant professor at Harvard Medical School and a faculty member of the Precision Vaccines program at Boston Children's Hospital, where she also leads the data management and analysis core. Her research focuses on characterizing immune responses to vaccines and infections using systems immunology approaches. We also have with us Dr. Juulee Thakar. She's an associate professor at the Department of Microbiology and Immunology at University of Rochester. Dr. Thakar leads a research program focused on understanding how the immune system responds to infections, vaccination, and environmental exposures. Her lab uses an integrative systems level approach combining bioinformatics, systems biology, and dynamic modeling to analyze complex immune data. So today's presentation title is the Immune Signatures Data Resource, an AI-ready compendium of systems vaccinology datasets. With that, I will just hand it over to Dr. Arce.
Thank you, Sanchita.
Okay, let me stop sharing.
Yeah, there you go.
Yeah.
Okay, one moment. Can everyone see my screen well?
Yes, perfect.
How about this? All right. Since Sanchita already mentioned the title of our talk, let's go through the next slide, which I really want to set this stage for the introduction, especially for the talk, which will also introduce import. And this is an import webinar, so it's very important that we understand about this resource. As you know and mentioned here, it's an immunology database and analysis portal, which makes it easier for sharing immunology research and clinical data. And what makes it more than just a repository is its commitment to standardization. As you can see in this diagram right here, that data deposited here are harmonized through a consistent data model, applying controlled vocabularies and ontologies across studies. And the goal is to move beyond siloed datasets and create a foundation that actually enables integrative analysis. The data flows from the providers, data providers like you, through quality assessments, the identification, especially of sensitive PHI, semantic harmonization, which ultimately becomes accessible to broader community. This slideshow actually shows us the 3 AI-ready datasets that Import developed as part of the NAIRR pilot. Sanchita mentioned what NAIRR is all about, which is the National Artificial Intelligence Resource Pilot, which aims to connect U.S. researchers and educators to computational data and training resources, especially right now to advance AI research. And the contribution of Import is very important as it develops and curates open and high quality immunology data sets ready for use by the broader community. Some of the examples that was already made available by Import. Let's start with the first one, which is the 10K Immunomes, which is a reference data set of immune profiles from over 10,000 healthy control subjects across 83 studies, serving as a baseline for human immunology. The second is a COVID-19 dataset, which is a harmonized collection of 22 curated studies across 2,200 subjects or participants, compiled to enable meta-analysis with data types such as flow cytometry, ELISA, neutralizing titers, and other mechanistic assays. And the third, which is where we will spend the rest of our time, is the Immune Signatures Data Resource, or what we call the ISDR, which is a compendium of standardized systems vaccinology datasets, capturing transcriptional profiles and antibody response measurements across 30 studies, 24 vaccines, 13 pathogens, and 1,405 participants. And this is what we will be walking you through in detail. But before that, I would like to briefly ground everyone to the scientific framework that motivates this work. And this is the systems immunology, as you can see in my slide right here. Instead of looking on just one gene or one pathway at a time, we take a holistic view of the immune system. We integrate transcriptomics, proteomics, metabolomics, or some sort of omics that you probably have heard about, and other high dimensional data. And the goal is to understand how these components interact as a network or as a whole, and ultimately to predict outcomes like what we're talking about today, which is vaccine responses, or even with disease outcomes or modeling. A major contributor to this field is the Human Immunology Project Consortium, or HIPC, which is a NIAID NIH-funded collaborative infrastructure through which much of this work is coordinated. And this curated effort, we have developed that particular resource, the ISDR, which is a compendium of system vaccinology data sets. And I'll walk you through how we are able to do that with this slide. So again, the ISDR, or Immune Signatures Data Resource, aggregates and harmonizes systems vaccinology data from, as mentioned here, a lot of these cohorts, 53 cohorts from 30 studies of 24 vaccines, 1,405 participants, and 4,795 transcriptional profile. And what distinguishes ISDR from just a simple repository is the rigor of the curation pipeline. Data were sourced in the HIPC centers, so you can see in my slide right here, through Import and GEO, such as the Gene Expression Omnibus. Then we've also searched up some of the systems vaccinology papers in PubMed. And we were able to process them through a multi-stage quality assessment workflow. And again, we're inputting those all in Import, and we utilize Immune Space for our virtual analysis. We process them through a multi-stage workflow, which includes some of the quality control methods that we have applied, such as array quality metrics, batch correction. We have also done age and biological sex imputation and normalization. Before it was being organized into virtual study ready for analysis. And as I mentioned, we have utilized Immune Space, which provides an interactive access to this data. And what these data sets actually look like is that it follows a longitudinal sampling scheme from pre-vaccination to post-vaccination. And I can show you later on some of the studies that we have, which even extends throughout the year, which is a temporal structure across which transcriptional dynamics are captured. This particular slide gives you a sense of the scale and the breadth of the ISDR. On the left panel, you can see the participant count broken down by pathogen, vaccine type, or assay modality, which includes transcriptomic, HAI, or hemagglutination inhibition assay for influenza. For those that are non-influenza studies, we do have also neutralizing antibody and ELISA, which probably captures, for example, IgG and other components. As you can see, studies of influenza's vaccine are highly represented, but of course, we're also capturing other studies as well. The bar chart on the right shows the sample counts at each of the time point post-vaccination, with the heaviest sampling at 0, or the baseline sample, and 137, which we know as the critical early window for innate and adaptive immune activation. The Venn diagram right here, you can see, illustrates the overlap across assay platform, and the partial overlap reflects that the reality of this multimodal immunology data, that not every participant has every assay at every time point. But this is where the harmonization pipeline is specifically designed to handle. One of the landmark analyses and papers that enabled by the ISDR is the transcriptional atlas post-vaccination we published in Nature Immunology in 2022. Here we identified a conserved blood transcriptional module centered on plasma cell differentiation and immunoglobulin production. We were able to consistently predict antibody response across diverse vaccine platforms. And this heat map shows the module activity scores across vaccine and time points, capturing the early innate signatures, as I mentioned, at day 1. As you can see right here, you can see the red panel here, or the red upregulation of those transcriptional modules, and also later B cell and plasma cell signatures emerging by day 7. Seeing the same transcriptional predictors across different adjuvants, vaccine, and pathogen really shows us what is possible when this data are standardized and harmonized. Next slide actually gives you a glimpse of what we're going to do next with version 2 of ISDR. And compared with the version 1, the resource has gone quite a bit. It now includes, as you can see right here on the panel below, 51 studies, 109 cohorts, and over 2,500 participants, with close to 10,000 expression samples and more than 8,000 antibody response measurements. We have also added several important vaccines dataset you can see right here, which will include SARS-CoV-2 mRNA vaccines, dengue tetravalent vaccine, bordetella pertussis, hepatitis B for additional cohorts. We also have typhoid fever, pneumococcus, and influenza data as well. And with that, with the background information I've given, I will hand it off to Julie to walk us through the next part.
Thank you, Joann, for a nice introduction to the data set. So what I would like to do is now walk you through the tutorial and some of the details of the resource. Our motivation for developing this ISDR dataset as AI-ready dataset was to improve the access to the data and also help automate the workflows developed for the analysis of this dataset. We believe that the AI-ready datasets will increase the analytic efficiency, data integration, and meta-analysis across these vaccine studies. We also would like to promote wider use of this dataset that will hopefully help additional studies. The work done here is part of National Artificial Intelligence Research Resource Pilot, and funding came from Office of Data Science Strategy. Next slide, please. So as Import started thinking about developing AI-ready datasets, one of the key challenges we faced was how to define AI readiness. since many different data sets are ready at different levels or have different preparation stages, and there are a lot of nuances when we think about these data sets. So Import came up with these 4 categories for defining AI readiness. The first category is data preparation, where we evaluate the level of the data ready to be used. So things like Joann mentioned, right? Batch correction, normalization, these factors are evaluated when we are looking at the data preparation. Then the next factor is data quality, where data is evaluated for its completeness, consistency, integrity, and provenance. Then we look at data documentation. So what level of metadata is available with this data set, whether there is a data dictionary, ontology, is data published, is there a DOI? So these factors are evaluated for the data set. And finally, we like to evaluate data access, which is the format, delivery, and usage of the data rights. So things like, is there any identifiable information? What security measures need to be taken to be able to access this data set? So as we develop Import developed these 4 categories, we use the assessment as we started developing the ISDR data set for AI readiness. Initially, some of these scores were lower, and as we developed the AI ready data resource, the scores were improved, and at this point, we have almost max scores for every category mentioned here. And later on, I'll show you how you can access this AI-ready assessment for ISDR data set on Import. So one of the things I want to mention, can we stay on the previous slide? So one of the things I want to mention is that the scores that are assigned by this assessment are subjective. It's a self-assessment, so they cannot be really compared across different data sets. Yeah, next slide. So now I want to dive deeper into the data set and the tutorial. So the tutorial was developed to perform the random forest classification on the vaccine response prediction. And on the left side, I'm showing you the subjects or the participants available in this study. So there are broadly two groups, you can say young individuals from 18 to 49 and older individuals from 60 to 90. The tutorial uses only young adults from age 18 to 49. And this data is used for further analysis. On the right-hand side, I want to give you a sense of how antibody responses look like. So for influenza vaccine, we can see how the antibody titers, log fold change of the antibody titers vary across the population or the cohort. Most of the studies in this data set have a study design as shown on the top right corner of this slide. So it has the baseline value or data, which is taken before the vaccination, and then there are time points available after the vaccination. And as Joann showed, these are the vaccines that are available in the data set, and they are shown on the bottom right of the slide. Next slide, please. So one of the goals of this webinar is to really help you use these data sets. So I want to talk a little bit more about the metadata that is available with this data resource. So of course, we have participant and study level metadata, which includes participant ID, study accession, ARM accession. So ARM accession is a identifier for study arm, clinical trial arm. So some of the studies can be developed in future to evaluate these data sets within the arms as well. And then there is cohorts such as young and older groups. So the second set of metadata is demographic, which is basically race, ethnicity, sex, and age. Again, as Joann mentioned, we have imputed age whenever the age is missing. And then we have vaccine-related metadata, which is name and year of the vaccine administered, vaccine type, such as live or attenuated or inactivated. And then we have pathogen that is targeted by the vaccine. And then there is assays, right? So assays measuring the immune responses. And for the tutorial, we are using HAI to define the response, but there are other measures available for everyone to look at, such as neutralizing antibody titer and ELISA, which has isotype specific or total antibody responses. And then there is also adjuvant. Whenever it is applicable, that is also described as in the metadata. Next slide, please. So how do we define our classification problem? So here we discretize the response between low and high. The responder group was defined, grouped into low or high based on maximum fold change across all the strains on day 28 compared to the baseline. So this variable is encoded as MFC P30. For the classification problem, as I said, we define low and high responder using the quantile thresholds specified by the 30th percentile cut point, as you can see by P30. But again, I mean, this is not the only measure, right? So you can use this data set for using other measurements also. And below, I am listing all the other measurements that are available. So max RBA is a participant's immune response classification based on maximum residual baseline adjusted response. Then we have max strain MFC, which is viral strain that produced the maximum fold change. Then we have max strain RBA, which is viral strain that produced the maximum baseline adjusted response. And any one of these can be used to define your responder and do the analysis. Next slide, please. So at the end, I will go over the tutorial, but this is to give you idea about how we visualize the results. We run the random forest and we plot the ROC curve. And then we also evaluate model performance using area under the curve. So on the right-hand side, x-axis is different vaccines and y-axis is AUC. So yeah, these two metrics are used for evaluating the random forest performance. Next slide. Okay, so now we'll look at how you can find the data resource and then go over the tutorial. Okay, so if you go to the import, this is the website you will land on. You can click on the resources, and then if you scroll down, there is this NAIRR AI ready datasets. And here you can find all the different datasets that Joann talked about. So 10K immunome, COVID-19 dataset, and ISDR dataset. In this dataset, here is the tutorial that I'm going to go over today. And the publication that Joann mentioned. And this is the assessment that I was talking about. So you can also look at how ready this data is and how we scored each factor in the assessment. And now I will stop share and show you the tutorial.
Julie, you want to take questions now? There's one question while you are
Sure.
setting it up. Is there any follow-up data available?
So this is the data that is available currently. The version 2 is not currently available, if that's the question.
Yeah, that is what I've already mentioned, that the version 2 is currently underway. So we'll just wait for that. And maybe we can do another tutorial like this.
So do you see my screen now?
Yeah, I do.
Okay. So this
I will
is
take the
the,
other questions later
okay.
after the tutorial. Okay, great.
Okay,
Go
let's
ahead.
do that. Sounds good. So this is the tutorial. Here, there is information that we just described early on. And then I'll just walk you through some of the steps. So first, we prepare the data. So the files are, the main two files that you want to use are all immune data with response.rds and norm ESET. So norm ESET has the gene expression data, and the response file has all the immune responses, all the metadata that I was telling you about. So basically, first we upload the data, and then you can look at all other metadata when you want to explore the data set. And then here we also filter the participants only to look for younger individuals under 50 years old. We merge the immune response and the phenotype data and create a combined pathogen vaccine type identifier for use of the analysis later on. Then we do basic data cleaning a little bit, retain only pre-vaccination sample points, because here we are using baseline to predict the vaccine response. We exclude the studies with fewer than 10 samples to maintain the statistical power, remove the genes with any missing expression, drop known outlier samples based on UID, and then we create the matrix for machine learning. And there is code available for you to look at and run. So as I mentioned, this matrix basically combines the gene expression along with metadata. So here we are using MFC50 to define define our response, low and high response. So then here we define the responders. We can again look at the code. We assemble the training matrices, identify the participants common to phenotype and gene expression data, select top 500 most variable genes for modeling. And then in this section, we are performing model training with 10-fold cross-validation. And here we will run the model with for random forest classifier. Training data is centered and scaled, and model is trained to maximize AU ROC, and parallelization code is provided for Mac or Windows. Here we extract 10 cross-validation predictions, training predictions, and feature importance score. So as I mentioned, we are evaluating a model using ROC curve and the AUC, so you can look at the vaccine type and accuracy, go over the AUC, and then we have some plots to look at the genes and their importance scores. So this is PCA for top 500 most variable genes. You can look at the predicted probability distribution by class, and I'm not going to go over detail. but you can look at, the genes that have high weights and variants. Yeah. I think I'll stop here. Feel free to go over the tutorial, and if you have any questions, please reach out. I'm going to go back to the slides now. Do you see my slides in the slideshow?
Yes.
Okay. So after developing this tutorial and the data set, one of the first thing we wanted to see is how can we use these data set directly in some AI tools like ChatGPT and Copilot, and what level of prompting do we need to develop the analysis. And this was really a short-term project, so we basically evaluated multiple different prompts to decide that, okay, two-step prompt works reasonably for initial basic analysis. So for ChatGPT, we uploaded the data set in zip format, all the data set, including metadata and the gene expression data, and developed two-step prompt. In the first step, we asked the ChatGPT to group the samples and then develop the random forest classifier specifically. And this is the outcome we got, which was reasonably good. So we did get AUC, and then on x-axis, I'm showing you the vaccines. However, more work is needed if we really want to change the grouping of the individuals and ask more specific questions, and that is not possible just by simple prompts. And then we tried to do the similar approach with Copilot in Visual Studio, where we group the samples, and then in this time we just asked to develop classifier. So Copilot generated 5 machine learning models, which had Elastic Net, Random Forest, SVM, XGBoost, and Neural Net, and produce this outcome. So here I'm showing you AUC in green, sensitivity in orange, and specificity in blue, purple. And then on x-axis, you see performance of 5 different models. And we can also look at top 30 features that were identified that had higher importance for these models. So this was basically quick try to see how the AI-ready dataset performs in these two tools. Of course, more work is needed to really ask specific questions. So that's it. That's all we have. And I would like to thank the team. So EAB, the External Advisory Board, for all the feedback that we received, NIAID team, UCSF team, ICF team, and the Paraton team, and of course, all the data providers. And also thanks to the funding resource, funding support, which came from NAIRR pilot through Office of Data Science Strategy. And I'll stop share and happy to address any additional questions.
Yeah, we can pause briefly to allow the questions to come in. Meanwhile, I can ask a question. Thank you, Joann and Julie. That was a great presentation with a lot of information in there. So I'm tempted to ask a question on like what kind of challenges you had while building these AI-ready data sets. Of course, there are a lot of opportunities that you have demonstrated. Did you come across any barriers or challenges?
I can probably mention that, especially in the early on when we're making the curation possible. Again, we're curating from 53 studies. What we have seen is, some of the studies were actually a little old already, and we were using microarrays on some of them, but we also need to harmonize it with, Illumina sequences. So we have to really make sure that they are consistent. We have done per study normalization before we even do cross study normalization. And what we've also seen, and as I pointed out, the biological index, the sex imputation, that sometimes when data providers are actually sharing their data and then they're inputting like whether they're male and female, some of them are missing information. Some of those are probably switch from male to female. Again, that's only some of the conclusions that we have done, but we really need to make sure that we are providing clean data, quality assurance data sets and metrics with that.
Yeah, thank you. Some of it we have also come across. So.
Yeah, especially import. Yeah, of course.
Right. Any other questions from the audience?
And also
Use
something
the data
that,
set. Go ahead, sorry.
Yeah, something that, you're really thinking that this is for AI ready. When we were curating this data, we did not think about AI, but it made our life easier because of how rigorous the assessments we have performed and some of the metrics we have done.
Yeah, those kind of documentation comes in
Exactly.
handy. Like
version control.
Yep.
Where are the codes? Did we even use the same code or same data sets? How do we filter them out? Those are very important.
Thank you.
Go ahead, Julie. Sorry, cut you off.
No, I was just going to say to everyone, use the data set and the tutorial and let us know if there are any problems, questions.
All right, so I think we have some time. I'll just take 2 minutes just for some important announcements and information. So let me share my slide. All right, do you see the slides?
Yes, yeah.
Okay, yeah. So many of you might be interested if this webinar is recorded. As I mentioned earlier, yes, the recordings will be available within few days or maybe even sooner. And if you are interested to go and check out the other webinar archives, here is the link. Go to Import website and type in webinars, and you could access the webinars from the past and this one very soon. Here's the QR code. Recording materials are also available in the YouTube format for accessing anytime. Connecting with Import, if you are interested to know more about the Import datasets, we host office hours on the first Thursday of every month from 2 p.m. to 3 p.m. Eastern. Discuss your questions directly with the Import team. And then we also have newsletters. So we release newsletters on a monthly basis, which highlights new data, resources, features, Import events. So feel free and register. Here's the QR code for newsletter registration. We also have a presence in the social media whenever there are new data sets or Import team is presenting at the conferences. To keep yourself connected, please feel free. We Import is on through LinkedIn, YouTube, and also on X. And we have tons of documentations and tutorials. If you want to learn how to access datasets, analyze datasets or from these public repositories, probably tutorials is another resource within Import that you can access, download it, and probably want to practice if you want. These tutorials are meant for for the starters, how you download. The codes are available in the Python format. We have a Jupyter notebooks that you can access and get started. So I'll just check if there are any. Meanwhile, there are questions. If not, then probably I'll just checking. Okay. Yeah, I think it was a great presentation and a great resource that Joann and Julie you have built, and hoping that the community can use it and leverage it for their own research purpose. I would like to thank the participants for your attention and joining us today, and hoping that this was valuable for you, and you're going to join us for the next one, and keep yourself engaged with this import webinar series. I think I was able to give quite a bit amount of time back in your calendar. Thank you very much. Thanks for joining again, and thanks for the presenters as well. Thank you.
Thank you, everyone.
Okay, so I'm going to close now. Thank you. Oh.